All right, so thank you for the invitation, Breathe and to Aware. I'm quite happy to be here today. I'm just going to say that I'm a scientist and not a psychiatrist. And so this is from a research perspective on the brain and imaging the brain and what we know about that so far. When you look at a scene like this, or if you've climbed, I mean, it's Ireland, I suppose I'm from Galway, so I've been a little biased here, but if you get that sense of exhilaration, one of the things that has puzzled me for a very long time is exactly what is the essence of that sensation, that feeling, the exhilaration or amazement. It's not just the physical climb or the view. There's obviously a lot that's happening to the body. And one of the goals I have here today is to open the black box a little bit about how our environment affects us. How does it get in? And how is it that we go ahead with life, we can see, we can hear, and we physically move our hands, and we sort of forget that the body's there, but it's doing an enormous amount. And I think it's part of a case for hope, because it's the mediator of those feelings. It's what generates the difference in your experience of some environment that might be quite different to someone else's experience of that environment. You've got two recipients of an identical environment or an identical conversation, but it impacts them very differently. They experience the same conversation very differently, and even differently at different times in your life. So the brain mediates that. And what I'd like to try to achieve is to open that a little and say how. Where are the physical connections, the actual anatomical bits that do those jobs to some extent? And that's in addition to pointing at the research and what the research knows about, what we know about bipolar disorder from scanning the brain specifically. So this is a brain, and I mean, I think it's a marvellous thing. I think it's absolutely fascinating. I thought nothing could be as exciting as scanning it or physically seeing it. I think that was because I was so fascinated with how remarkable the job it does is. And when I first started, one of the first lectures where someone introduced me to the brain, I was just absolutely grasped by it, and I couldn't leave it ever since. And I mean, I look at it and it is just a blob of jelly, and I was quite obsessed with it for a while, and discovering all the little bits and what each bit does and so on. And we've really, I suppose, moved away from that a considerable extent in research, from saying, well, this is the part that does this job, or this is the part. And one of the points today is that the future research that's going to benefit psychiatry specifically is that the brain doesn't work bit by bit. It isn't modular. You can't take one bit out. It all works together. Some of the parts of who you are indivisible from some of the parts that cause you trouble. Sometimes, not always, but sometimes you are part and parcel the parts that perhaps you aren't as happy with at times, and other times are. Yeah? So physically looking at it in isolation would be folly, wouldn't it? It's just a brain on its own. Thinking about it and how it interacts with the environment then becomes much more important. So this is the brain. And there are some places that actually do a very specific job, like some frequencies of sound are processed in exactly one tiny little bit right here, and the next frequency up is here, and the next frequency up is here. And this is hearing. And so some parts are quite specialised, but complex behaviours, like the ones that we tackle in psychiatry, they're very much a network of regions that interact with each other, and they influence each other. You can seed one and say, to what extent does that influence the activation of other areas? And you're looking at patterns then about areas that are connected with each other. So it's very much the research would be moving away from a sort of a location by location approach. I think location by location came about a little bit more because we couldn't look at it as a whole in 3D even 40 years ago. We were perhaps stuck with post-mortem investigation and taking out tiny bits and looking under a microscope, whereas now you can get into an MRI scanner and do something, interact with an image, emotional or non-emotional, and we can look at the pattern of activation in different areas at the same time across the entire brain. So it's now feasible to say, well, which areas are actually working together in this network to mediate behaviour. and they're all affected by each other. I suppose I'm going to come back to one example, so I might mention one. I'm trying to connect up the brain and environment a little bit non-physically, but I'm going to do it physically as well. In terms of your sleep as well, that affects components of the networks that are important for emotion. So all of the advice that you all generally get does have a physical basis. There's a meaningful physical basis for needing to be active physically, verbally, having conversations, sleeping well, eating well. It sounds terribly simple, but there is definite neuroscience demonstrating the effects of sleep on the brain, and sleep can change how you process things, not just the speed of them, but change precisely how those networks function. And so they're all critically important and have a scientific physical basis as well. So this is me, and this is my favourite thing to do, to hop on a scanner. And so this is me standing like this, with see the long nose just at the bottom there, right? And so there's the frontal lobe of my brain, and there is the back. This is the cerebellum, and here's my brainstem going down to the spinal cord. And you can kind of see a little bit of the area where smell happens, just at the very top of the inside of the nose. And so this is a standard MRI. This is a magnetic resonance image. And it's just structural information. It just tells you about the shape of the brain or the parts. And it's very static information. It's just shape or structure. But you can do lots with an MRI scanner. You can look at the function of the brain. You can ask someone to do absolutely nothing in the scanner and just lie there. And it's called resting state functional MRI. And you're accessing the parts of the brain that are active when someone isn't doing anything, but you're never really not doing anything, are you? Sometimes you're thinking to yourself, so you're introspecting. And so there's a network for introspection or self-thought that we now know, and we can describe the parts that are involved in it. And one of the key regions that I'm excited about telling you about, the cingulum, is a core part of that emotion network. It's part of this default mode network, the network that's used when we're not really doing anything, when we're just thinking to ourselves. And so it's important that that network has time to be active. When it's active, we're not. It can't be active at the same time as we're concentrating or paying attention to something else or doing a task. So you have the two systems, and sometimes you're involved in a task, you're paying attention, and one network is active, and other times you're not, and there's another network active. So from this kind of structural image, you can see some bits that look a bit whiter and bits that look a bit more grey. And we call this the cortex. And of course it undulates in and out, up and down, and there's lots of different parts. And in the grey, it's what we'll term as cafes. It's where you go to have a chat. So the grey matter, we're going to talk about grey matter and white matter, and the whole thing is made of grey and white. The grey bits create the surface all around the outside. It's everything you're looking at on the outside here. And they're the cafes. They're where one neuron comes in, has a chat to the next, and passes the message on. And the message, of course, then they're mediated by different chemicals. So the chemical could be dopamine. You've probably heard a lot of these, so I'll throw a few out, but dopamine for reward. If you're thirsty and you have a drink, dopamine is released. There's serotonin, more to do with mood, and acetylcholine would be another one. I don't know if you've heard of it, but it's one of particular interest to me, and it mediates attention. So you're releasing it as you're paying attention, acetylcholine. And in the earlier days, I suppose, of neuroscience that was relevant to psychiatry, there was a very strong focus on these chemicals, to the point where a lot of the targets that are used for medication focus on these systems. We don't necessarily mechanistically understand them. So when I say what I'm hoping to talk a little bit about are the development of safe, effective, evidence-based treatments, I don't mean evidence in the removed sense of we think it works from a clinical trial. I mean a mechanistically understood treatment. So I want to step back a little bit and spend some time understanding the brain so that you might devise strategies for finding targets and medications that are based on knowledge and not necessarily serendipity or accidentally coming across particular medications. So that's the grey, they're the cafes, so you're in there for a chat and the dopamine's talking to another dopamine, or the dopamine's talking to a serotonin, and they go on a bit. But there's these conduits, like a ride-on bus that they have to take to get to the next location, the next cafe. So they had their latte, but they need a tea, and they're going from A to B, and it's the conduits are the white matter. So connections, basically. And along those conduits, we ignored them for a long time too, because on this type of MRI, which we've had for about 40 years or so, they all look just white, don't they? We can't really tell which direction these bits are going. We don't know which grey matter bit they're moving from and which one they're going to. And that's the scan that I'll talk a little bit more about today. It's an ugly old head, isn't it? To kind of show you how real the physical basis for this is. This isn't maybe just looking at feelings. This is trying to connect the feelings to actual data in a scanner. To make sense of that, I wanted to spend a minute just on this slightly grotesque image. It's a remarkable feat of dissection of the human nervous system. And the brain is just this tiny little triangle right up there, and these are all the nerves in your body. And it's the nervous system that mediates a lot of what we've just talked about, and everything that is relevant in terms of getting messages into the body, but also the decision making that occurs inside the body, and then the things that result from that behavior or movement. So the brain is just there, and then these are nerves for the head and neck region. So they might be sensation to the cheek, or they might be muscles to the tongue. And then around your neck region, the nerves that come off that part of your spinal cord, they generally go to your arms and help you move your arms. And then there's some nerves that come off in the middle, and they go to the thorax and help with muscles of breathing and other boring things. And then a lumbar region, go down to the legs. So there's a general organisation to it. And they're all relevant to us because the brain is the core centre and level at which conscious experience of feeling, be it physical or otherwise, and activity and so on, are mediated. So they can all be accessed, and that's a part of the opportunity that we're in some extent missing a little. They can all be accessed via the body externally through the nervous system. This is a weird looking little diagram up here, isn't it? This is called a homunculus. And this is one half of the brain. It's as if I'm standing like this, and we just cut straight through here. So coronal slice. And it's just looking at one half. And it's specifically looking at a piece of cortex that mediates how you feel things. So sensory, so just incoming things. So if you feel a touch to the forearm, there's a particular piece of cortex that receives that and senses that you just had a touch to the forearm. And the reason the diagram looks all skewed in size is because it's to represent the amount of cortex that's dedicated to particular physical parts of the body. So your entire lower limb, big and all as it is, proportionally, only gets that much cortex there. Whereas, look at the hand. It's a huge piece of cortex in comparison to the entire lower limb. And the face and the tongue. And that's, of course, because we do so much more in terms of dexterity with our hands. There's vastly more specialization for the tongue and the hand and so forth. So this is how these signals are getting in. And they all have a physical path. They come through a relay station, and they go down through the brainstem and down the spinal cord and out, or in, out in terms of motor, or in terms of sensory, from every part of your body, head and neck and so on. And I suppose at this point you're thinking, well, am I just talking about walking and moving? Well, no, this is the very point, I suppose, that the large research grants are looking for to fund research now in psychiatry. It's that we take account of the environment. Taking account of the environment simply means allowing for all of the other aspects that are relevant in psychiatry. Not just looking at the brain and not just looking at clinical symptoms, but bringing the two together and bringing together what affects it. So, for example, certainly incoming sensory information that you'll all be familiar with are the eyes for vision or smell. And you have nerves that come off the back of the eye and they move into the brain, and then they integrate into the little cafes and have a chat. So they're in the cortex at the very back here when they're processing what you've just looked at. So vision. So that's directly accessing cortex in the brain, and it's activating in response to seeing an image. And if that image is emotional content or non-emotional content, that may have slightly different network that it goes on to activate thereafter. The amount of time we're talking about, say, if there was a red apple sitting there and I looked at it, the amount of time might be less than a second for you to say, well, yeah, that's a red apple. But what's physically happening between the point at which the image of that hits your retina and reaches back to this primary cafe back here, the visual cortex, and then relays out to some other region where you kind of go, oh, I think they taste good, yes, they're food, and integrates it with all the other aspects that helps you recognize it as an apple. It all takes less than a second. It might be, say, 600 to 800 milliseconds, but in that space of time, it can have visited 12, 13 pieces of cortex very, very quickly. So it'll bounce from one to the next and back, and recognition and integration of what you're thinking about as you recognize it is barely perceptible, but it's happening. And so there's communication with the environment, and you're not always aware of every step in part of that communication or environment. So if you're spending cognitive time socially figuring out what is written on someone's face, and you're trying to figure out, well, is this a positive vibe or a negative vibe I'm getting right now? You might perceive all of that, but it's still happening. It still uses networks and is occurring while you're processing things. So we have the grey, which are the cafes, and the white, which are the conduits or the bus to get from the different cafes where the communication's occurring. And I mentioned that we have ignored the white or the conduits or the routes to some extent in terms of research for quite a while. Up until about 20 years ago, they were mostly just described from postmortem studies, whereas now we can see them in vivo. That's vastly important because now you can look at them while someone's well and while someone isn't well. And that's a huge step forward than, say, just looking afterwards. Are these intact? Are they present or absent? Are they different? Now looking at them at different phases during different times for people makes it quite different. So this is what the cafes look like. There's one neuron and there's another, and they might be having a chat. And inside each neuron is the nucleus, and inside that is the DNA when they talk about genes and genetics. So every one of these has DNA, and whether or not one bit of that is read or not, it differs, it varies a little. And so one of these could be one chemical like dopamine, and another one could be serotonin, or they can be the same type, and then they communicate with each other. So that's where the communication's actually occurring. They're all wrapped in myelin. I don't know if you've heard of myelin, but if you've heard of MS, you know it's a disorder where myelin is disappearing, and the velocity, the speed or efficiency that electrical signals move down neurons is impaired. It slows down, so their ability to control their muscles as a result is slightly impaired. This is wrapped, you can see it wrapped in circles here, a little bit like one of those big industrial irons where you pop the sheet in and you just keep turning the handle and it just wraps around and around. And so of course you need lots of layers of that, and you need it nice and tight and organised, and you don't want things in the between and bits missing. And that's effectively a kind of an insulation. And in these little vesicles inside you have the dopamine or the serotonin. And you may have more of that or less of that, and that may be as a result of a medication or your environment or the genetics, and you may have a limited capacity to release. We all have a limited capacity across time. If I tell you you've just won €1 million and I tell you again in a minute, you just won't be as excited the second time. You haven't, right? When you eat the first bite of pizza, it tastes really, really good. The third or fourth, not so much. So we have, physically, that when we feel these things, they are the sense that we have that these chemical things are happening. And there is a limited capacity in there. So they're the communication zone. So these routes between the cafes, the white bit, they just all look the same when you dissect a brain postmortem. You can't really tell in here whether this one is running up here or whether it's running over here. So we've been really restricted in how to understand it because of that. Similarly, in these normal, general structural MRI, you still can't really tell. But now with a kind of a diffusion MRI, which is a little bit like looking in at different angles. So instead of just taking a snapshot, we can now, they're a bit tricky. They're railroads, right? They're like the track that this conduit or ride-on bus takes. And if you look down the track in line with it, you can't really see it. But if you look in at the side, in fact, if you get exactly perpendicular to it, you can see it quite well. And so what's important about these scans is to take a look from all the possible angles you can. It takes a while, it's very noisy, but by looking from all different angles, you can map them out and see exactly which directions they're sitting or going in. And so here's the track, for example. This is a really high resolution and this is microscopy, it isn't in vivo, but I'll show you some in vivo data as well, and you can see exactly where they're going. So this is pretty exciting, because now you can take the data that we already know a lot about where the cafes sit and exactly what they're doing, and now you can start to infer about the connections between them and how they're talking to each other. So I wanted to put in one point a little bit about how important the tracks are in terms of development. So that you're not born with a lot of that myelin, that sheath, the insulation we talked about. That comes as you develop, and it begins to build in the teen years and the early 20s. And you have a very different way that the brain works across that time, as you know, if you know any 4-year-olds like I do. Okay, so you can see the brighter it is right here, the slightly more organised and ready these bits are to work. And so back even at 1 week, what is it that a one-year-old baby's doing physically and with their environment? They're just sitting there kicking. They don't really do much else, do they? So this is the corticospinal tract. This is a bit that goes and helps big, gross movements in the body. And so that's one of the first things. And of course, their interaction with an environment, putting little socks with bells on and them hearing that, and then they discover they can move it, and encouraging that activity helps with this development. And you can see the amount of white, how bright it's getting here, is getting brighter and brighter until even at 10 years, there's a huge difference, isn't there, from 1 week. So there's critical windows in the development, both embryologically before they're born and even all the way up to sort of the 20s as this changes dynamically, during which there's a risk. You could call it a vulnerability, you can call it an opportunity, but it's either or. So there's a development trajectory for this white matter itself. And what's great about these scans, where we look in at all the different angles to see which way they're going and exactly where they're going, is that we can kind of color code them and start to describe their direction. So in red here we have things going from one hemisphere to the next, and in blue we have things coming in sort of the direction from your feet to your head, and in green things from the back to the front. So some of the things that I've read that other people have done with this that I find pretty fascinating are looking at children's brains and saying, well, yeah, the front part of their brain, the frontal lobes, they're really well connected with themselves, and the middle is connected well with itself, and the back is well connected with itself. But actually, when you're an adult, the back is also very well connected with the front. And so you can start to see, if you subtract these two maps, this is what's different in a child versus an adult, apart from not making any sense at 4. You've got large difference in how connected distant parts of the brain are. So there's a trajectory across time for how someone might be able or capable of processing certain information. And the timing of that is delicate and beginning to be reasonably well described. So I've mentioned a bunch of different things that affect your brain. I've mentioned genetics, and I've mentioned the brain itself interacting with the environment. And if anyone really still harbours any doubt that the environment can affect the brain or brain development, which I doubt, but just in case, here is two CT images of a normal 3-year-old and a 3-year-old who was exposed to extreme neglect. So they're the identical age, and this neglect is social, emotional, nutritional, everything. Very, very serious. And brain development is very different. You can see larger ventricles, that's the space inside that just contains a fluid. It's not functional, so you want those to sort of, they usually become smaller with time. And you have the skull on the outside. Can you see that the child's head is in fact smaller? So all of the growth curves that the PHN does, they're relevant in terms of growth, and so you can see a huge difference. So there just isn't any doubt anymore that all of the aspects of who we are and our environment affects the brain. And in turn, therefore, the brain has a given capacity to deal with the environment. So that changed the capacity to deal with it. And that is, again, it's a vulnerability, but it's an opportunity because it's dynamic and it changes all the time. So in other words, as you live in an environment, you are changing your brain by dealing with things and interacting with the environment. So I suppose it's both. An example that is, it's very early stages, but by giving someone a training test where you show them the level of brain activation they have in response to something and asking them, can you change it? Can you do this task again and reduce it? Can you do it again and reduce it further? There is some preliminary evidence that in fact by giving them the tangible image of their own brain activity, that they're in fact able to modify that. And that's very sort of early stages, but it would have enormous potential. In terms of, when I last checked a couple of years ago, there were only about 20 or 30 studies looking at the effect of psychotherapy on the brain. And you can clearly see in response to psychotherapy changes in energy usage in the brain. And that sometimes leads on to the question, well, can we not therefore then use it to look at diagnosis? I don't personally think that's useful or necessary. And I'll try to explain why I think that over time. But someone at the beginning raised the question, well, there are websites in the States that claim that you can have a brain scan that would diagnose, and they asked me to maybe comment on that. And of course, that's not real. It's hugely hopeful, but it's not true and it's not real. They look at energy usage in the brain, and there isn't the data behind it to genuinely diagnose. And why would you want to anyway? Would it actually help? So one of the points, I suppose, that I'm getting to is that, in fact, they're more useful if we could predict which treatment was more likely to be useful for a given person, wouldn't it? We have psychiatrists, and what we need to do is add to that and make it additional information or tools for them to use. So certainly environment, from the point of view of psychotherapy, having a positive environment, so knowing when certain environments are creating a negativity around thought versus, say, a positivity around thought, that's your environment. Situations and scenarios that cause negative feelings or that trigger them are part and parcel of what I mean when I say environment. I don't just mean looking at the red apple. That was more of just a physical example to show you that they all genuinely come in and activate brain networks themselves. So this is my problem with psychiatry today on video. For the sake of determining exactly what medication someone should take, the biological tests that are done are to rule out other things. They're not directly examining which, say, for example, medication might work better. They're not directly going to diagnose, and they can't yet, and I'm not sure that's really that important. So, for example, you could have in the wonderful DSM, or Diagnostic Statistical Manual, individuals who fall into any one of these three different diagnostic boxes, so bipolar type 1, bipolar type 2. But depending on their symptoms, the medication will be chosen based on their symptoms. So you have an antidepressant. So two of these different boxes could have some depression, and an antidepressant will be used. All three of them could have psychosis, an antipsychotic will be used. Two, but maybe not the other, may have an anxiety involved, and then an anti-anxiety medication is taken. So unfortunately at the minute research has been hampered by taking these a little too seriously, by taking the categorization of bipolar disorder type 1, which personally I believe is multiple biological entities. It isn't a single thing. And the reason that it's a single thing is because at the minute the only way we can figure it out is by asking questions and looking at the family history, looking at the personal history with no biological tests. So that's where I think this research fits. It fits at the cusp of attempting to introduce those biological tests. To cover the parts that are possibly the most exciting, the highest funding, the bits that people around the world are feeling extremely hopeful about in this area, I wanted to step out to two huge projects. And one is a little bit of a kickback to the Human Genome Project. Back when they mapped the human genome, they looked at DNA and they said, we want to know every letter in sequence for the human genome. 3 billion letters in a row. It would fill a huge tome of a book. And they code for all of the bits that you have that work, that dopamine we talked about for the reward and the first bite of the pizza, not the fourth. To code for, say, dopamine, for example, to make it 3 letters make one piece and another 3 make another piece, and it all goes together to finally create something like dopamine, that chemical. And of course, we all have a lot of variation in our DNA, and one of the main queries in psychiatry is, well, is it because of our gene? Is it because of my genetics? What I would say about that, from my own experience, and I'm going to recommend a book that goes into hugely more depth, is absolutely fascinating, called The Gene, and I'll put it up at the end, is that It's a blueprint, it's a book, it's what's read to create the parts, but again, the environment influences, and this is more recent knowledge that we know. In other words, it can be tightened up in some locations and then it's not read, or it can be loosened out in other locations and then it's read. And if you remember that image of the two neurons I showed you, each one has a copy of all of this DNA. So in one brain region, it may in fact be open and we can read them all, and in another it may be closed and we can't read them all. And our experience is exactly what we're exposed to can change that. So hopefully I've unstuck any notion that genetics is just a book, you're stuck with it and it caused everything. It can't be true, because it's an interactive book. It's something that you're changing through your life experiences. In fact, we think, and this is only from animal experiments, that in fact the experiences of your parents or your grandparents may in fact have changed your book to some extent. The bits that tell it whether it can be read or not the actual letters. So there's a lot of changes in that area about genetics that are hugely fascinating. They can now take one gene and change it. It's extremely highly regulated, and it's only useful in very simple diseases where one gene causes the disease. One gene codes for one enzyme, that enzyme's missing, you need that enzyme, they might change the gene which then creates the enzyme, now it'll work. But to scale that up to the complexity of psychiatry isn't quite there yet. It's exactly what's been funded and been worked on. And we are trying to take the genetic information and combine it with our information on the brain from imaging, and combine that with our information from the environment to attempt to look at the interaction between the genetics and the brain itself. So what's going on right now in the world of imaging is that they've realized that we have this huge map of the genome, and we know the gene and the brain interact dramatically closely. We don't have that beautiful map of the brain. So, if you have one dictionary and you don't have the dictionary in the second language and you're attempting to interpret a passage, how on earth would you? So we need a map of the brain. Similarly, we have one tiny worm that only has 200 neurons mapped completely. It has 200 neurons and they create 7,000 connections, and we have a map of every neuron, every connection, every cafe and every route between the two cafes. It's a worm. Scaling that up isn't possible yet because we haven't got a scanner that's so powerful that it's like looking down a microscope in your brain. Instead, what we have are scanners that can look at hundreds or 1,000 neurons in one little clump and tells us information about these little clumps. So the technology is going to hold us back a little bit, but we now know how to do it. We know how to do it mathematically, and we have it on some scale. We just don't have it on a neuron by neuron scale, and that's a bit future. That's a bit of the future. Brain scanners now can do enormous numbers of different things. You see this it's boring to you now, right? This structural MRI. Now we're getting a bit more excited. This is functional activation in response to doing something. This is a molecular PET scan, so this is a little bit like, if you've heard of Prozac, obviously, binds to the serotonin transporter and it blocks it. And the transporter, you can do a scan and see how many transporters there are in different regions of the brain and start to look at that when someone's well versus, say, when they're depressed. So there's scans for specific neurotransmitters as well as just structural information. And then these conduits are routes between different places. And of course, brilliant images of the vessels as well that are used in stroke. So the Human Connectome project is the equivalent of the Human Genome Project, where they tried to map the entire genome for a human. They're trying to map the entire brain for a human. It's not neuron by neuron level. It's a slightly different scale. But you can see these conduits, and these red ones, they're traveling from the right side to the left side, connecting the brain. And in blue, we have things that are going down the spinal cord. And in green, see these two little ones here, green, this is the cingulum bundle. This is a key part of the emotion network. And we can see in bipolar disorder that it's not as organized as it is in a group of people who are not experiencing symptoms of bipolar disorder and have never had any episode either way. So this cingulum bundle, it's connecting different emotional parts of the brain, and it's a particular network called the limbic system. So I'm going to pop out to a video that you might have already seen because it's on the BBC News site. So this is not too far away, but I want to point out one thing as it swings around. So these are the conduits or the connections. This is the white matter in the brain. This is like we took off all of this cortex, the grey matter cafes on the outside. We took them all away and we're just looking underneath at the connections from one to the next. And this is done in, can you see that little green one just in there? It'll come around again and we'll look in. That's the cingulum. And this is done in Cardiff, where there's only 3 scanners that are the best resolution. There's one in Cardiff, one in Leipzig, and one in Minnesota. And they're very exciting to me. I nearly moved there. They do this. It's the best resolution we have of these connections. And as you look inside, so it's just coming down around it and looking at all the different connections in different places. Sometimes it's giving you a green colour where they're fanning out into the grey. And other times it's just trying to show you organisation of one lobe compared to the other. So it's quite remarkable, and even though it just says dementia, epilepsy and MS, we are using it in bipolar disorder as well. So in the centre of that, maybe you noticed, maybe you didn't, that cingulum, this connector of the emotional system. And so here it is drawn out on its own if we just take everything else away and track it out. So again, this is a view as if I'm standing like this, where this is the front, there's the top of the nose, and there's the back of the head. And so here you have a connection bundle that joins the right and left hemisphere, and just above it we have some cafes and conduit called cingulate cafes and cingulum conduits, connections. And the chatter that goes on there is very, very much about our internal emotional state. It's very much about interpreting the emotion in the room around us, or how exactly we feel about something that's happening at that time. And it integrates from multiple locations, sometimes more strongly and sometimes more weakly. It integrates from places that mediate fear or anxiety, like the amygdala, and the connections aren't necessarily as strong. So in particular, if I took, so here I am again, I jump into the scanner anytime I can, and if you took, say for example, a slice like this, and you decided to just make a dot out of each cafe, yeah? So every dot, this is a connectome, this is what they're attempting to do, to create the connectome map of the brain. And each dot is one of our cafes. Now, you can see that they're big and they're spread out, and that's because we're not neuron by neuron. This is an entire brain, and here's the front and here's the back. And we're looking down at the top of a head in this view. And so here are each cafe, and the strength, the thickness of these lines is how often it's connected to the next one. And so you see a general pattern. And so we do images like what I just showed you that track the white matter, and we do these kinds of structural images to define where the cafes sit, and we put them together to create that connectome map. And then some mathematicians get very excited, and this is where I think it's in its infancy, and it's a little bit early to get terribly excited about exactly how we're measuring it and what we're going to be able to tell from it. But, for example, for something to get from A to B, if you remember the example of looking at the apple and things going through different regions of cortex, let's say it's a non-emotional situation, but because something just happened, you're still processing things in a slightly different way. Maybe you take a slightly more circuitous route where you're trying to evaluate the emotional context of a decision as opposed to just making the decision without that emotional context. The path that you'll take for that information transfer will change. If you drop this node and you go a long route instead, you've increased the path length, maybe the time, maybe the processing time for getting from A to B. If we take that and we try to compare, so what you're looking at here, again, it's just that network, the connectome map, and it's rotating, which hopefully helps and isn't, maybe it doesn't help, maybe it's just confusing, but you can see that some of those nodes are bigger or smaller, and I've just weighted them to be bigger if that region's more connected to other regions compared to others. So you naturally see that there are parts of the brain that are more connected and parts that are naturally less connected, and that's true of everybody. So if you took any 2 nodes and asked how many steps it needs to take to get from one cafe to the next, and you did that from all possible cafes in the entire brain, averaged, you get a measure of that number of journeys possible. And you can see, so each dot is a person that was scanned, and these do not have bipolar disorder and these do. They're mostly bipolar type 1, and you can see that the spread is different. There's more of a spread, but you can also see that plenty of these individuals are just the same as this control group. But on average, as a group, they're slightly higher. So there's no line I can draw here and say, well, if your path is longer, you have bipolar disorder or you don't. And that's why I suppose I was saying that there isn't a scan that I've seen at all that can do that can differentiate or diagnose. And again, I'm not sure that's really what's important here or what's necessarily useful. So it's an average overall that's different. Similarly, if you just look at the parts of that connectome map that do the heaviest lifting in the brain, in other words, they're more connected to themselves than they are to other parts of the brain. That's what we call a rich club. Funny old name for it, but it's a set of regions that really do more talking to each other. Bankers, maybe.
Rich club.
The rich club, yeah. So it's these set of brain regions, and if you statistically compare a large group of people without and with bipolar disorder, you can see that there are some locations, like the cingulum, that isn't accessed as often in the group of individuals with bipolar disorder. But it is by some of them. But on average, it's less so. So there's a lot of information in that about exactly what that might mean. And that's the cusp, really, of where the papers, if you get right down to the discussion parts of the most modern research articles possible in psychiatric neuroimaging, that's where they're struggling to explain that at the minute. So the maths is very well developed, the technology is very well developed, the resolution it will take more time and needs to improve. But these are some of the things that are being done. Of interest, what the Rich Club does as its day job is information integration. So if you look in a large group of people at this Rich Club, what is it actually doing? It's integrating information from multiple sources. That's what it does. That's the heavy lifter component of the brain, if you like. So what would be my hope for this kind of work and the kind from the study that we might talk a little bit about at the end, is that we would develop knowledge or tests, perhaps in a more translated way, that would define groups that individuals can move into that have a biological basis that would then be used to determine which medication could be pursued more readily or maybe more likely to work. That's where I see a real hope, not necessarily in diagnosing, but in categorization. Why do I think that would be important? Because I think it could potentially reduce the time spent unwell. And I think we can only possibly do that by integrating across those levels that I've talked about. I mean, I've talked about genetics and those letters that make up the gene, and they're really, really small scale. And they influence the chemicals like dopamine. And then in the cafes in the grey matter cortex, those chemicals like dopamine, they talk to other neurons. And then they go down the tracks to other areas. And maybe they can't if there's an impairment on those tracks to talk to other regions. And it scales all the way out to the environment and how the environment affects us. And the environment, again, can affect our cognition and behaviour, you know a lot about. It can also affect that neuroanatomical network, the connectome map. It can also affect molecules, and it can affect genes. And again, if you've any doubt about the environment affecting molecules, there are scans looking at that serotonin transporter that Prozac targets, and it changes with daylight sunshine hours. So those molecules vary naturally in our body depending on daily sunshine hours. Good bit better in Canada than here. And similarly, we also have examples where the environment now, especially supported by animal research, that the environment in fact can directly affect whether the genes are going to be read or not. So there's a lot of interaction across all the levels of scale. And you can see the complexity of the problem. And hopefully you can see a little bit about how that physically functions and works in the body. So my hopes for where this kind of research and some of those little things I told you about what's different would be that we look before and after treatment, for example, and say, well, what exactly did that treatment do physically in the brain? Or predict treatment response, which is more likely to work based on something physical about that person's connectome map. And similarly, but this group of ideas over here, because these are all ideas that are run in clinical trials. These are the different designs that the research is funding. They might affect your quality of life sooner if you're a patient, but they all depend on a treatment already existing. So looking at that treatment. And so they're a little bit restrictive if they were the only thing we did. And so you've got this other packet down here that is obviously going to take a lot longer to in fact affect anyone's life. But they have the potential, in my opinion, to identify new targets or new strategies for treatment, pharmacological or otherwise. And so these more challenging designs, I think they're more longer term vision, and they're a little different, but they're inherently further away from making a difference. So I think it's therefore essential to pursue both, and you see that a lot in the funding that happens. So this is where I go to think, and this is, I suppose, what the research is attempting to do: combine information about the brain with genetic, clinical, cognitive and environmental factors in bipolar disorder to try to make a meaningful contribution to the science of what we understand about this. It's a little, so it's a message of hope in the sense that you can see from some of those data that we're on the cusp of having tests that could be useful. But we can also see right now that they aren't actually in use, which is a real shame, and that I think is what's changing at the moment, and there's fairly dynamic discussions about those changes happening around the world. So here's some of the things that I think you would really enjoy if you've enjoyed this material. This book is by Siddhartha Mukherjee, an oncologist in the UK who is about genetics of oncology, but he has a family history of psychiatric disorders, and he writes this book about the gene from the perspective of someone with a family history of psychiatric disorders. It's phenomenal. I thoroughly enjoyed it, and it's on the discussions about it on Twitter are saying that it's more than just enjoyable to scientists, that it's genuinely accessing a lot of individuals, and it's fascinating. And he goes right through to his futuristic vision of where he thinks that genetic research might go in psychiatry specifically. So that I strongly recommend that. It's on the Wellcome Trust book prize list, so there's no doubting that that's a good resource. This is a really old one, one of the first that sort of intrigued me. Again, completely understandable. It's philosophical in its angle, much more so than scientific. So if you're more interested in the philosophy of how the brain works and how we understand feelings and thought, John Searle is a relatively elderly philosopher who has an incredible take on it. And he also has talks on YouTube and TED, which are, again, completely comprehensible and do a lovely job of trying to talk about how feelings manifest in the brain and how much we understand about that right now. This one, maybe you've all already read it, but again, it's a neurologist and psychiatrist's point of view on having bipolar disorder herself. Therefore, it's incredibly insightful because it's from a clinically informed, medically educated person with bipolar disorder. So a lot of people who will have read that one would attest to its insight. And then this is Sebastian Seung, S-U-E-N-G, apologies. And this is the connectome. Again, this is written for the public, but this is less so about psychiatry and more so about the potential of the connected brain to inform us about how the brain functions. So a little bit more on the science side. And again, he also has TED Talks, S-E-U-N-G, TED Talks that are very accessible and easy to listen to. They're 15 minutes and they tell you about the excitement around the connectome. It's a little oversold, but that's TED. Thank you very much for your time.